ये YouTube में नहीं आ रहा क्या? YouTube पे आ रहा है?
क्वेश्चन आएगा
Okay. Hello everyone. This is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and welcome to the live session of Enzyme Science and Technology. And I think we'll start with the introduction itself. So, Ishan, can you introduce yourself? Ishan, can you turn on your camera and introduce?
Sorry for the camera setting, sir. I'm in my living room. So, hello, sir. My name is Ishan Bennett. Hello?
Ishan? Yeah.
Okay. Hello, sir. I'm Ishan Bennett. I'm an undergraduate student. I'm pursuing my 6th sem in zoology. And I've taken the course to learn and explore more about science, and I want to do my master's. So I'm searching for more courses to select which stream I want to take in my master's, or which specialization, things like this, sir.
So from where you are doing the BTech?
No, I'm not doing BTech. I'm doing Bachelor's of Science. Yes.
From where you are doing?
B. Barua College, Guwahati, sir.
Oh, B. Barua College. So what are the subjects you have?
I have my main subject is honors in zoology. And till 6th, I had till 4th sem, I had chemistry as my second.
Oh. Do you have any specific question?
Sir, for specific questions, I don't have, sir. Sir, I have taken the course and I have still, and most of my doubts have been cleared by the forum, sir.
Okay, fine. So, do you so far you have submitted all the assignments?
Yes, sir.
Okay. So I think 5th assignment is going on, right? Or
5th has been submitted. I think it's going.
Okay, fine.
6th.
So what we are planning is we'll have a brief overview of whatever we have discussed so far, and that may actually remind you some questions which you can ask.
Oh, sir, can I turn off my camera now, please?
Yes. Don't be nervous. Okay, yeah, turn off the camera. Yeah. यहाँ से शेयर होती है ना. Here we go. ठीक है ना, कैमरा ऑफ हो गया ना? मेरा चेहरा आ रहा है? Okay, so welcome to the live session 2, and this is just to give you a brief overview what we have discussed so far, and then I hope it will start the discussion about the content of the course also. So what we have discussed so far, what we have discussed is that basic introduction about the enzymes. So as you know that the enzymes are made up of proteins, except that the ribozymes, which are made up of RNA, the proteins are the enzymes are present in enzymes are present in all the living cells, and and they are converting the substrate into the product. ये पुराना प्रॉब्लम स्क्रीन दिख रहा है, एरो दिख रहा है.
Sir, arrow and screen are visible. अब नहीं दिख रहा.
Wait, I think they are trying to sort out something. It's writing is not visible actually. Huh?
Now it's okay, sir.
Is it okay now? Can you see the writing?
Yes, yes, we can see.
So we said that enzymes are present in all the living cells, right? They are made up of the proteins, except the ribozymes, which are made up of RNA, right? And you know that the ribozymes are very specifically having a role in the splicing, so that we have not discussed in those in this course anyway. Enzymes are converting the substrate into the product, and many times the enzymes are running the reaction in both the directions, right? So they can actually be able to convert substrate into product and product into substrate, right? In most of the cases, the reactions are reversible, right? And these are biological. They are also been called as biological catalysts because they are. And what is mean by the catalyst is the substance which will increase or which will enhance the rate of reaction, but it will remain unchanged, which means it was not going to be utilized in while the reaction is going on, which means your enzyme is not going to be consumed during the reaction. So how the enzymes are catalyzing these reactions? So enzymes are catalyzing the reactions by lowering down the activation energy, and so you can imagine that if you are running a chemical reaction and if you have two reactant A plus B, and when they are reacting, they are forming the C plus D. So these A and B are actually having the different types of bonds, right? So you can imagine that if I have AOH plus B phosphate, right? Then what will happen is that this bond is going to be broken down. This bond is also going to be broken down, and then this particular group is going to be present here, and this will go here, and that's how you are going to have the A phosphate plus BOH, right? And this is going to be called as C, and this is going to be D. So it happens because the A plus B are actually going to interact, and there are so many bonds which are going to be broken, and new bonds are going to be formed. And you know that the breaking of the bond is requiring the energy, whereas the formation of the bond is always been associated with the release of energy, right? So this is actually completely going to be governed by the energy, right? So if the A and B are present at this particular free energy, right? And if you require a product, right, they are supposed to cross this particular energy barrier, right? And if the reaction is uncatalyzed, then they require more energy, because then you require to break these bonds with the help of the energy what you're supplying. But if the enzyme is present, it is actually going to lower down this energy, and this energy is nothing but the activation energy, right? So this activation energy is going to be lower down, and as a result, because you don't require much energy, right, you will be able to run this reaction at a faster rate. And that's why we need an enzyme in the biological system, because of the simple reason that a non-enzymatic reaction would be in the scale of is going to be several hundred folds enhanced when you are actually going to run the same reaction with the help of enzyme. Classical example is carbonic anhydrase, right? So carbonic anhydrase is an enzyme which is going to break the which is going to form the carbonic acid. So if you have a non-enzymatic reaction, the reaction rate constant would be in the range of 1.3 into the power minus 1, whereas when the reaction mechanism rate would be in the range of 10 to the power 6. So there will be a rate enhancement of 10 to the power 6 times, which is actually close to 10 lakh times. So that's why you are actually requiring the enzyme for catalyzing the different types of reactions. In the same module, we have also discussed how the different types of enzymes are being classified, and we discuss about the enzyme commission. And what you can see here is the enzymes are being classified into 6 different groups based on the reaction what they are catalyzing, and all these enzymes are assigned as a enzyme commission number, right? So this is the way you are actually going to provide the enzyme commission number. And I'm not going to discuss all these in detail. And then we have also discussed about the nomenclature, how you are going to give a name to an enzyme, and what it says that the rule 1. So rule 1 says that you are actually going to utilize all the trivial names what are being used. For example, you are going to use the enzyme which is called as pepsin. So these are the trivial names. So they are still being used as such. But apart from that, you are always going to use the substrate, and where the substrate is normally in the form of an ion, its name should be end in the 8, whereas if it is rather than ik, right? And the enzyme name would be always be by the name of the substrate. So for example, if you have the enzyme substrate as lactic acid, so it will be lactate, and the reaction what it is going to catalyze is an oxidation reaction, so it's going to be called as lactate dehydrogenase. And rule 3 is the commonly used abbreviation for the substrate. For example, the ATP and all that can be used, but you cannot use the new abbreviation. Rule 4, for example, if the name of the substrate composed of 2 nouns, then it should be placed in with the name of the hyphens. For example, glucose 6-phosphate 1-dehydrogenase. So you have the 2 nouns, so it's going to be separated by a hyphen. Then the rule 5 says that use of the name of the enzyme description, such as condensing enzyme, pH of enzyme should be discontinued as soon as the enzyme catalyzed reaction is known. And in some cases, when you are actually unaware of the final acceptor, like for example, in some of the oxidoreductase reactions, you don't know who is actually going to accept the electron, and then you are actually going to write within the parentheses, you are going to write the acceptors because you don't know about the acceptors. And the rule 7 is systemic name should be given should have the 2 parts, name of the substrate and the process specifying the name ending with the S. So we have already taken a few examples and we have discussed in detail about these nomenclatures. You all can stop me whenever you think that this is actually something which you should understand or you should want to discuss, so that we'll have a fruitful discussion. Then we also have the classifications and all that. So that anyway, we are not going to discuss. And this is just an example of class 1, which is oxidoreductase, right? And then module 2, we discuss about the enzyme structures, and we said that the enzyme structure is having the four different folds. We have a primary structure, secondary structure, tertiary structure, and quaternary structures. We discuss the different types of methods, what you are going to do for the primary structure, secondary structures, tertiary structure, and the quaternary structure. So for the primary structure, we discuss about how you can be able to determine the protein sequence or the amino acid sequence, right? And then secondary structure, we discuss about the CD and IR spectroscopy. And the tertiary structure and the quaternary structure, we discuss about the experimental method and as well as the non-experimental method. So this is just an overview of the protein sequencing, where you are going to take the protein, then you are going to react. First, you are actually going to denature the protein. You're going to destroy all the disulfide linkages and all that you are going to do with the help of the reducing equivalents. And then you are going to react this enzyme with the Sanger's reagents, and you are going to hydrolyze the amino acid, and you are going to separate the amino acid, and that's how it is actually going to form the 2,4-nitrodenyl. For example, if you have the first amino acid as glutamate, so it is going to form the 2,4-dinitrophenyl glutamate adduct, and then this adduct actually is going to be separated, and so on. So after that, you can actually be able to cleave this protein with the help of the trypsin, and then you can actually be able to do the degradation. You can actually be able to do the sequencing of the individual peptides. And since these peptides will have the overlapping sequences, you can actually be able to put them together, and that's how you can actually be able to get a complete sequence of the whole protein. Then we also discuss about the protein structure determination methods. So we'll say that there will be two approaches. One is the non-experimental approach, where we have discussed about the homology modeling, and then we also discuss about the experimental procedures. So within the experimental procedure, you have two conditions. In condition 1, suppose you have the protein for which you have the crystal, then you can actually be able to perform the crystallography, X-ray crystallography, right? And you can be able to determine the structure. If you don't be able to get the structure, or you will not be able to get the crystal, then you can actually be able to do the protein in the solution. You can express the protein, then you can actually be able to perform the NMR spectroscopy. And either of these approaches are actually going to give you the final structure, although there are several differences between the structure, what you are going to get from the X-ray crystallography versus NMR spectroscopy and so on. And this is what we have discussed when we were discussing about the homology modeling. So, and I'm sure you might have seen a video where the student have discussed about the different steps. So in the all these steps we have discussed in detail about, and we have also shown you a demo where we have shown how you can actually be able to use the modeler, and how you can be able to use the modeler to model the proteins. Okay. And if you want to study more about this, you can actually be able to go with this particular reference, and it is actually going to give you the complete detail about the homology modeling. And then we also discussed about the protein production, because for any kind of applications, for any kind of utilizations, you need to provide the proteins, right, in large quantities. So when you want to do the protein, first step is that you are going to identify the gene which is corresponding to the particular protein. And once you identify the gene, you are actually going to clone that particular gene. So identification of the gene depends on whether you are looking for a protein which is expressing. So in that case, you are actually going to prepare a cDNA library, and you can actually be able to use the cDNA library, and you can screen the your gene into the seed in the library. Similarly, if you are having the protein in the genome, but it's not expressing, then you can actually be able to prepare a genomic library, and you can actually be able to use that. And either of these libraries, you can actually be able to use different types of screening method. You can use the enzymatic method, you can use the DNA-based screening methods, and so on. And so once you got the gene, then you can actually be able to use the different types of cloning techniques. So you can actually be able to do the clone with the help of the gene amplifications, cloning, and screening. And this screening is different from this screening, right? This screening would be for your clone of the recombinant DNA, okay? And once you got the clone, then you can actually able to put it into the protein production. So you can actually do the overexpression. So you can actually be able to select the suitable host. Host could be of two types. It could be a prokaryotic host, or it could be a eukaryotic host, right? Within the eukaryotic host, you can have either the yeast, you can have the insect cell lines, or you can actually be able to have the mammalian cells, and you can also have the virus, so virus-based transfection. So once you select the host cells, accordingly you have to select the transformation method. So if you select the prokaryotic system, you can actually have the chemical method or the electroporation. If you select the eukaryotic cells, then you can have the chemical method. You can have the lipofectam. So you have different types of methods for transformations. Once you got done with the transformation, you can actually have to do the screening, and then you are going to get the desirable clone. And once you got a desirable clone, that you are actually going to use for overexpression. And once you've done the overexpression, you are actually going to do the analysis into the SD. Okay, so you are going to check the expression into the SDS page. Once it is confirmed that you are getting the expression, then you are going to put it into the over purifications. And the purification, for purification, first you have to devise the strategies to disintegrate the cells. So you're going to have the cell disruption methods. So you can have the mechanical method, you can have the enzymatic method, you can have the other kinds of physical methods, and so on. Once you have done the cell disruption, the cellular content is going to be released, and then you are actually going to have the chromatography methods. You can have the post chromatography method. You can do the estimations to know what will be the yield of the protein, what would be the purity of your protein, and so on. And at this stage, you can also do the enzymatic assay to know whether the enzyme what you have purified is active or not. And then you can actually be able to do the purification analysis to know whether the product is good enough for further downstream applications or not. At this stage, one of the students has asked one question. Okay, and this is a very interesting question, and that's why I have taken this into the live sessions. So what he was saying, and this is something which is related to the screening of the recombinant clone. Okay, so screening of recombinant clone. So what it is saying is, and it is about the negative selection. Okay, so I'm sure if you want, you can actually be able to go through with the content. He already given the link actually, and this is I have taken from the discussion forum. So what it's saying is that in a negative selection, you have three different types of species. One is the host cell. The other is host cells plus vector, which means does not contain the recombinant DNA. And the third is host cells plus recombinant DNA. Okay. And the approach is that you have a vector which actually has the gene, okay, which has an enzyme, and this enzyme is producing. So this is actually producing an enzyme, and this enzyme is converting a non-toxic product into a toxic product. Okay, so if this enzyme is active, which means if you this particular full gene is available, It is actually going to produce the toxic product, and it is actually going to kill the transform cells. Now, this question student has asked what, so if this is the case, the host cell, which actually contains only the vector, are actually going to get killed because they are actually going to produce the active enzyme. Whereas in the case of this, because it also has the recombinant DNA, and your recombinant DNA, what you are going to put, your recombinant DNA is somewhere here, right? So because you are putting the recombinant DNA within the enzyme gene, it is actually going to produce the inactive gene. So once you have the recombinant gene, it is actually going to produce the inactive enzyme. And since it is actually going to produce the inactive enzyme, these cells will not be able to convert a non-toxic product into a toxic product. And as a result, these cells are actually going to survive. Now, what he asked is what will happen to the host cells? Okay, because host cell does not have any sensitivity to this or any kind of method to this. So how we are going to screen actually the host cells and this cell? So for this, actually, you are actually going to do the double screening. Okay. Now imagine that this is the all the genes, what all the cells, what you're going to get. Okay. And for the just, I'm showing the big one as the host cells. Okay. Now, if I transfer this onto a membrane, I'm going to get a replica of this, right? So I'm going to get a replica of this. So if I compare now, okay. So if and so now what I'll do is I'll transfer this, right? And I'll add the compound which is going to kill, right? So there are so many cells which are actually going to be disappeared. The only cell which is actually going to be remain on this is the host cell and your cell, right? Now, host cells, because they do not have the recombinant DNA, their size is actually going to be bigger. That is first indication that these are the non-transform cells. Second is their ability to survive into this particular media is actually going to be less. And the third is you can actually be able to put them onto a antibiotic screening. Okay. So if you put them onto a antibiotic screening, what will happen is that these host cells will actually going to die. Only your cells are only going to survive. And this is what you are going to get finally the screen cells. Okay, so you are going to get the screen clones, and that's how you can actually be able to use them for protein production. If you do this antibiotic screening here itself or at any other stage, then first of all, the antibody screening is not going to be very fruitful. The second is antibody screening this vector is also going to give you the resistance. So it will actually going to survive. Okay, so this is the way you should actually be able to screen out the host cell by doing a double screening. I hope it is clear to the RIA general actually. Now we also subsequent to that we have also discussed about the enzyme catalyzed reactions. So we discussed about the catabolic reactions, we discussed about the anabolic reactions and so on, and we discussed about some of the important reactions from the catabolic. So we discussed about the glycolysis, and we discussed about the Krebs cycle, and we discussed about the beta oxidations and so on. Within the anabolism, we discuss about the amino acid biosynthetic pathway. We have also discussed about the urea cycle and so on. Okay, and the idea of discussing these reactions is that just to show you the importance of these enzymes into the different types of metabolic reactions. We also discuss about the how you can be able to study the enzyme substrate interactions and so on. So we discuss different types of techniques like the ITC, difference spectroscopy, and so on. So this is all we have already discussed, right? And then we also discuss about the ITC, and this is what the basic principle of the ITC, and we have shown you a demo also, how you can be able to measure the heat evolved or heat absorbed while you are doing it. So you are going to have a reference cell, you're going to have the test cells. So this reference cell and test cells are going to be placed into a jacket, and this jacket, the temperature of this vessel and temperature of this vessel is going to be remain onto the same temperature. What will happen is that when you add the ligand is actually going to cause the remodeling or the rearrangement of the bonds, and as a result the temperature either will go up or temperature will go down, right? As a result of this, the temperature in this is also going to be changed. So there will be a change, there will be a difference in the temperature between the reference cell and the test cell. So as soon as that happens, the heater is actually going to turn on, or heater is going to be turned off, okay? And as a result, they again will come to the same level. So this is what exactly is going to happen. So as soon as you inject the ligand, the temperature will go down, right? And then again it come back, right? Again it come back to the vessel level, right? So in this process, what happened is that the enzyme, the machine is actually warming up the sample. And this warming up will actually going to be lower down as the time will pass, because you are adding the ligand, more and more ligand. So initially, suppose you started with the 100 enzyme molecules, right? So if you add one molecule of ligand, it is actually going to cause a change, right? And that change is going to be very high because one ligand is changing, right? And it is coming back, right? Again, you're suppose adding the 10 ligands, again, you're adding 10 ligands and so on. So continuously, it is actually going to be keep saturating the enzymes, what is present inside your vessel. And as a result, what will happen is that initially, it is actually going to be very high, and then it is actually going to saturate like this. Okay, and then you are going to get a saturation. After this, there will be no change because there will be no enzyme available to bind the ligand. So using this, you can be able to calculate many kinetic parameters. You can be able to calculate the dissociation constant. You can be able to calculate the stoichiometry in which the enzyme and the ligands are interacting with each other. You can be able to change the enthalpy and so on. And then we also discuss about the different types of enzyme assays and so on. And so this is all about what we have discussed so far. We'll actually going to have another live session, and that time we will actually going to discuss some more about some more about the remaining content. So now, if I'm sure this could have given you some starting point to start the discussion. In case you have anything to ask, you can ask me.
Sir, this is a little bit out of course, but sir,
can you repeat again?
Sir, this question might be a little bit out of the content of the course, but
you don't have any question?
No. Sir,
Okay,
I'm about to type the question.
So Ishan, what you want to do? You want to do a master, huh? Ishan?
Yes, sir. Sir, my connection is very, very bad, sir. So, hello.
Yeah. Understand. No issues. Okay.
Sir, I'm about to type the question. Is it okay, sir?
Okay, fine. Ria, do you have any other question? I could see that you are live on the YouTube. Ishan, do you have any question? No, right? You don't have any question.
Sir, am I audible now? Sir, my connection is very bad, so I cannot like.
Yes, you are audible, no issues.
Okay, sir. So this question is a little bit, sir, out of the content, sir, but regarding the crystallization of the protein, sir, is the method very similar to the crystallization of the salt?
Okay, crystallization of salt.
No, is the method like we crystallize salts by like making a concentrated solution and then evaporating the solution, right? Is protein crystallization similar to?
Okay, so protein crystallization and the salt crystallization is same. The basic mechanism is same, except that the protein are little more, delicate molecule. Proteins are actually having the larger amount of interacting atoms. So it is difficult to predict under what condition the protein is going to crystallize. But because any salt what you take is going to have hardly 20-30 atoms, right? And so it is easy to predict how many types of rotation is possible, what are the different types of rotational freedom is there, what are the different possible interaction possible, and accordingly you can actually be able to predict that under what condition the salt is going to crystallize. Whereas in the case of protein, the protein, even the smallest protein is going to have approximately 3000 to 5000 atoms. So when you have the 3000 to 5000 atoms, their energy minima, where actually they are going to crystallize, is very difficult to predict. And that's why it is almost impossible for any software or any kind of algorithm to predict the protein crystallization. And that's why I think I have said, right, that's why you are actually going to try out different types of precipitant molecules, different pH, different salt combinations, and different pH and all that. Because you don't know under what condition the enzyme is actually going to adopt the minimum energy conformation, and as a result the protein molecules will start sticking to each other, and ultimately they are actually going to give you a crystal. Whereas in the case of salt, since the interactions are less, it is very much feasible. You can actually be able to crystallize any salt compared to the protein. Is it clear, or you were asking something else?
No, sir, it is clear, sir.
Okay, fine. So I think we don't have any more person to interact. We have already having the Ria on the YouTube. So I think whatever she was asking in the discussion forum, we have already answered. So since there's no more people who are actually available for discussion, I would like to conclude the live session. So thanks for your joining to the session, and I hope that you will join the next time also, when we are actually going to have a live session just before the exam. And that time, I think you can actually be able to, it's better to clarify all your doubts so that you will be well prepared for your exam, which would be in sometime in April actually. So till then, I think, yeah, bye-bye. And hope you will do very good in this course also. Thank you.
Thank you, sir. Thank you, sir.